Welcome. Here we are, all at home, safe, I hope. I hope the families are safe. Every member of the house knows not to go out and be congregating with very many folks. As being microbiologists, I'm sure you can appreciate that. We have a couple things to go over just to get us situated for the rest of the semester. I just want to assure you that I'm here to help you through this. We will get through it. You're going to do well. Hopefully we can have fun along the way. It will be different. We won't have the luxury of being together, which I miss, but it's something that we're going to have to do. So we just face it, move on, and so anyhow, let's do that. Let's do that here. So the to-do list for us at this first time that we get together in this modality is our to-do list really is going to be a simple one. I want us to get us started. I have a few things to go over. You can follow those. I assure you, you can always email me, and I have some contact information. You can even call my cell phone if you need to. But we also have Zoom and a few other things that I'll talk about for our office hours if you need that. So the to-do list for Chapter 6 is go through this presentation I'm working on with you together right now. And what we have is a worksheet. If you could fill that out using Word and just email it to me, and make sure that you get I received it response so that you know that I got it. And I like to do that until I get all the snags worked out. There may be other ways to submit the work, but for right now this will work for our purposes. Now you always have a week from the first day all week, so you can view this video at your leisure during the week. I have to break this one up into two pieces just because of YouTube's limitation. But and the other is that it is two classes. I am supplementing the time that we're normally together for 50 minutes. And so I try to cover all of the timing so you get your money's worth and still cover the materials that we need. And this will be modified as time goes on, or tweaked to everyone's satisfaction, I hope. And at least that's my goal. In lessons, all the Chapter 6 materials can be found, so you just click on that. There's nothing really new, and just migrate to the Chapter 6 and you can see the list of various things. One of the things that's not listed, of course, is this presentation, because I'm working on it as we speak. So that will be there for you. I'll also include video for lab today. I'm working on that. We really only have 2 labs to cover. The staff lab, which I took pictures of all your plates, and they could be found in the Staphylococcus lab. So your teams get together, I guess, just by finding your table number and your data. And it's one of the uses for teams is to find teams that is Microsoft Teams, and students will have access. You can work together on teams as a lab bench group and complete the assignments the best you can. Now I understand Teams is new and the whole format's new, so of course I limited the work pretty straightforward with the lab manual, and to do the staff and strep and the plates and everything are available. All I'm asking for you to do on the strep plates is these are your other section plates, and just find a representative for each of the numbers 1 through 4 and see if you can identify each of the unknowns as 1 through 4, and with the information I provided. That's it for the lab, but I want you to get used to working together on teams and then submit the lab report with the labs, which I will provide in Word format, so you can edit those easy. I'm working on that right now. So at least you can read through the materials and see what's done. By the end of the day, I'll have all the things reduced to Word format for you. So that's pretty much that. How to access Teams is listed in lessons. Just go in how to access Teams. I have 3 videos in there. The first one is how to access Teams for students, and that was written by Wake Tech administration, so you can look at that. And then I have 2 generic ones on how to use it and take advantage of some of the really nice features. They turned on Teams for you with all the access and bells and whistles, so it's free, of course, and they've included it, and how to access it is right there in the first video, so you can learn to do that and apply that for your labs and breaking up into your teams and for your bench as you have normally. Let's see how that works out, and I'll monitor it. I will be around various times on Teams if there's questions about that, or you can just drop me an email. And as I say, my expectations right now, let's get so that we get familiar with some of the tools, and we'll get through this. The tools I'm trying not to be overly too much technical. I want the easiest to use, and that's why I selected that. Zoom, I'm going to reserve to use that initially, at least for my office hours. You can see face-to-face, you can come up as a group. Seeing how the network, what I'm worried about is my bandwidth at my home. I also have a wife who does Wake County Public Schools, and she is constantly on the network. I call her the network pig. But so right now I want to limit the Zoom for our regular just office hours, but I may open it up for just a regular class meeting, perhaps, if students want that. It won't be mandatory, of course. It would be just if you want. Now, I'm also looking at creating the discussions by a new program, or it's an open source program. I'm using Camtasia right now, but I'm thinking about another one that streams and also produces a video. And if I give you the time at which the stream starts, you can watch that and interact with it if you'd like. That I'm working on. I think that would be a good feature, but we'll see. I've got it working. There's a lot of benefits to it, but for right now, we'll just see how it goes, and we'll go from there. So that's that. So if you have any questions, you can always email me, just like always. And then I gave you a phone number. That's my cell phone number, and feel free to call. If you absolutely, at last resort, have to get a hold of me. Don't put it on bathroom walls. Don't take it around or any place like that. This is just between you and I, and we'll go from there. So, Chapter 6, Microbial Nutrition and Growth. This is a pretty straightforward chapter. It's not exciting, I have to warn you. It's one of the necessaries. We have to understand about microbial nutrition and growth before we can really kind of get into the real understanding of how we manipulate and control these organisms. So we're going to look at the nutrients that are essential. We're going to differentiate between the terms. Again, it's vocabulary: macronutrients and micronutrients. List and define the 4 different terms that describe organism sources of carbon. Microbiology lingo, carbon source is the sugars usually that we add to the media. So what's the carbon source? Well, they use the carbon source for energy. We're going to talk about saprobes and parasites, and again, these are terminology type things. We're going to talk about stuff you should already know about, but I'll go over it again: diffusion and osmosis. We'll talk about isotonic, hypotonic, and hypertonic type solutions and how that affects the cells. And of course, that addresses the salt concentration or the exposure to salt in the environment and how microorganisms try to deal with that. 2 types of passive and active type of transports. Again, this should be stuff that you're already familiar with. So this is a pretty straightforward chapter. It's a good one to get started into this new sort of format that we're working on. and you try to keep everything consistent the way this is kind of rolling out. And I'd love to get your feedback. Let me know if this works for you. Ultimately, it's for you. So the microbial nutrition. Essential nutrients, any substance that must be provided to an organism to grow it. I mean, these are the things they have to have in order to do the business. And some of it they can manufacture on their own, but there are things that they absolutely have to have. Macronutrients are required in relatively large quantities, like water and that sort of thing, and plays principal roles in cell structure and metabolism. So these include carbon, hydrogen, and oxygen. The micronutrients are smaller amounts, can be also looked at as trace. These could be manganese, zinc, iron, nickel, various smaller functions, but absolutely required. If you don't include that, they're not going to grow. Inorganic nutrients are simple molecules that contain a combination of atoms other than carbon and hydrogen. So those are your inorganic. Found on the Earth's crust, bodies of water, various things. Usually we're talking about metals and their salts. Magnesium sulfate, ferric nitrate, these are salts of these sorts of compounds. Sodium phosphate. Or gases like oxygen, carbon dioxide, those sorts of things that easily go through membranes. Organic nutrients contain carbon, hydrogen atoms that are usually the products of living things. Simple organic molecules such as methane, or if you're from the UK, methane. Large polymers, carbohydrates, lipids, proteins, nucleic acids. That's the sort of thing that you should already know about the large polymers. Of course, water is an essential component, 70% of all components. Proteins, organic compounds make up about 97% of the dry weight. Carbon, hydrogen, oxygen, nitrogen, phosphate, and sulfur is CHOMPS is the elements, is a acronym to remember that. Most chemical elements are available to the cell as compounds and not as pure elements. The salts, the sodium versions of various things and that sort of thing. Only a few types of nutrients needed to synthesize over 5,000 different compounds, which is amazing. These are lean, mean fighting machines, and they know how to do it. Okay, some more vocabulary. This is talking about what bacteria eat. These are heterotrophs, organic. It's an organism that must contain its carbon in an organic form. So heterotroph has to go out, so it's different. Troph means it has a predilection or a needing for. Hetero is different, so it's different from itself or outside of itself. Autotrophs, an organism that uses inorganic CO2 as its carbon source, and they usually refer to that as carbon fixation or CO2 fixation, and they can actually pull it right out of the air. Has a capacity to convert CO2 into organic compounds. This is a really important thing. Now, we already are aware of those sorts of things, as in photosynthesis. Plants can pull CO2 right out of the air, incorporate that carbon into the plant, and bacteria can do the same. Not nutritionally dependent on other living things, which an autotroph, that's a really nice feature to have. A phototroph is a microbe that actually photosynthesizes or photosynthesizes. Photosynthesis is a really cool process, and photosynthesis is something that bacteria do. Chemotrophs are microbes that get its energy from chemical compounds. Okay, so there's some charts that summarize all of these things, and you can see autotrophs just with more detail. We have the chemo-autotroph or chemo-organic autotrophs. They use organic materials. And we have chemo-autotrophs or inorganic compounds. Chemolitho-autotrophs are minerals. If you think of lith as like a stone or a rock, that kind of indicate minerals, if you want to use that to remember. They refer to these organisms as rock-eating, so they're the lithotroph, and rock kind of works. These are pretty tough organisms. Some of these organisms that are in this group, they can survive on amazing things that we normally would say were toxic to us. Photoheterotrophs, sunlight, of course, purple and green photosynthetic bacteria. Chemoheterotrophs, these convert metabolically nutrients from other organisms, protozoa, fungi, many bacteria, and animals, of course. Chemoheterotrophs metabolize organic matter of dead organisms, and the fungi and bacteria kind of fall in that group. Chemoheterotrophs, or the parasites, utilize the tissues, fluids of living, various parasites, pathogens, bacteria, fungi, protozoa. In some cases, they still use leeches to. They only eat dead material, so it's one way of clearing up or cleaning tissues that are rather dirty and dead, maybe from a disease or something like that. Now, it sounds a little rough, but it's still used today. So the autotrophs are those that use photosynthesis. Autotrophs, that they don't need anyone else. They can get all their energy needs from photosynthesis, and they produce organic molecules using the CO2 that they can now pull from the air in the process of photosynthesis. Chemoautotrophs use organic compounds for energy, and they get that from their environment. The lithoautotrophs rely totally on inorganic materials, requiring either sunlight or organic nutrients. And these, again, they dwell at the bottom of oceans and really rough places. Chemoheterotrophs derive carbon and energy from organic compounds, process these molecules through cellular respiration or fermentation. Saprobes are free-living that feed on organic just matter or detritus from dead organisms, decomposers of plant litter, dead microbes and various things, and they recycle. These are the great recyclers, and we'd be up to our keister in dead things if we didn't have these. Parasites derive nutrients from the cell or tissues of the living host. Pathogens cause damage, which this is really kind of what we're talking about in this course as far as business goes. They cause damage to tissues or even death, range from viruses to helminths. Ectoparasites live on the body. Endoparasites live in the organs and tissues, which would be really nasty. Intracellular parasites live within cells. Now, viruses are obligate intracellular parasites, and there are some other organisms, even E. coli at times can be intracellular parasites, but Borrelia burgdorferi that causes Lyme disease is an obligate intracellular parasite, as others. So obligate parasites unable to grow outside of the living, leprosy bacillus and the syphilis spirochetes are additional ones to my list listed here. So there are several examples, many examples in fact, that are obligate intracellular parasites. Central nutrients, carbon, I don't need to really go over that much. These you should be familiar with. Hydrogen, usually to satisfy the octet rule, to make the compounds more stable with all the binding sites that they have for the octet rule, if you recall from your chemistry. So you look at the oxygen, it needs 2 more to satisfy all of the available electron seats, and hydrogens can do that by sharing it, so H2O, various things like that. These gases are both used and produced by microbes. Hydrogen helps maintain their pH. So again, there's duality in a lot of these things, the way they use these to maintain the pH or ways to live in their environment. Oxygen, major component, such as carbohydrates, lipids, nucleic acids, nothing new there. Oxygen is likewise a common component or inorganic salts. That forms the salt. That is why we call it a salt, is that it contains an oxygen type molecule. Phosphates, nitrates, and water. Free gaseous oxygen makes up 20% of the atmosphere, which was provided, by the way, by the bacteria friends that are in our environment. And so, anyhow, 20% of our atmosphere is from them. So, not bad. Nitrogen, major reservoir of nitrogen, is nitrogen gas, which makes up 79% of the Earth's atmosphere. It's indisposable of our favorite friends, the proteins, DNA, RNA, ATP, those sorts of things. Some bacteria and algae utilize inorganic nitrogenous nutrients like NO3, NO2, NH3. A small number of bacteria and archaea can transform nitrogen into other compounds. And regardless of the initial form in which inorganic nitrogens enter the cell, it must be converted to NH3. So the only form it can be directly combined with carbon to synthesize amino acids and other compounds. Okay, so it's pretty straightforward. I have a summary in the Chapter 6 directory that helps summarize all of these items that I'm going over, but they are straightforward. This is what I consider it's a great chapter to get us going into this new modality that we're in. It's straightforward. It is reviewed from some things that you already know, but it serves as a good way to get us back into the game. So phosphate and sulfur. Phosphate is inorganic source of phosphorus. PO4 is derived from phosphoric acid. It's found in rocks and various things. It's everywhere. Also used in ATP. Now, ATP, as you know, is the energy molecule. It's all about the phosphates and the double bonds and the energy that's in those chemical bonds that drive all of life. And so that's something. Bacteria. We talked about ATP synthase and the turning of the motor to make ATP. It's amazing, all coming from the basic element of phosphate. And so we use that for lots of things. Other important nutrients are potassium, sodium. Now we know a lot of these in muscle function, but these are for sodium pumps, driving energy types of needs. Also, it's important for certain functions of bacteria. Now, we talk about the pathogenic ones and ones that cause disease, and sometimes they steal our sodium and magnesium or calcium or iron, and they have to outcompete our systems for that in order to cause disease. So it's important to at least mention that. Now, zinc is another one that's essential for regulatory elements. These elements have to be associated with proteins. The protein by itself with the amino acids are incapable of pulling off whatever the function it is that's needed. So these are sort of the building blocks to make complement protein function. As we know, a good example, heme. Heme uses iron, and heme holds the iron molecule, so they work hand in hand together, and that's the same sort of thing here. Also, it sets up situations where we can get electrical currents and things like that. The microorganisms that we're not worried about as far as disease actually can be charged like batteries. They actually have, they can detect north and south magnetic various things using components like this. Okay, how microbes eat and transport mechanisms. Now we're going to cover this diffusion, and then I'm going to stop and break it presentation into a section or part 2. That way I don't get in trouble by YouTube. And also it gives you a chance to break and read through, replay certain sections if you need to. Again, I'd love your feedback on the quality and various things, and I'm trying different ways to do this. Online is not my favorite mode of doing things, but hey, we do what we have to do. So transport of necessary nutrients occurs across membranes. Usually this is what we're talking about. They have to get things into the cell. Not everything, things that they need. And we do this through a permissive nature of cell walls and embedding proteins and various things that facilitate the movements across various things. But one that we're going to talk about doesn't require any specialized hardware. It's taking advantage of just having that phospholipid bilayer membrane, and we have things that diffuse, which is a phenomenon of molecular movement in which atoms or molecules move in a gradient from an area of higher concentration to an area of lower concentration. and you can substitute the word density in there if you want, but concentration seems to make more sense to me. Now, osmosis is the diffusion of water through a selective or differentially permeable. In other words, it's not permeable to everything. Some things don't move in, some don't. This is osmotic pressure. And so if we have a lot of things that take up space or are devoid of water in one area, we have to have somewhat of an equal osmotic pressure. We're going to talk about that through a process of osmosis. Organisms that can't handle or maintain that osmotic pressure, they won't survive. They'll either shrivel up or they'll get so much water that they'll burst. It's the same thing that happens with our red blood cells. We have to have 0.95% salt in our blood. If that differs in one way, or let's say you just were to inject someone with pure water, that would be deleterious because the red blood cells, of course, maintain a certain concentration of water, and you have pure water on the outside, then you're going to get a net movement trying to make the two equal. So some of the salts will leave, or the materials in the bacteria will leave and it'll shrivel up, and we'll talk more about that. So this will continue until the concentrations are equalized on both sides, and that's the key to osmosis. The osmotic pressure, by the way, is driven by gravity. And so here is a container looking through a selectable membrane sac. You have solute and water. Solute takes up space or displaces water. And so when you have a pore size, which are permeable to the water but not to the solute, and you can see that what's going to happen is you're going to try to equalize the amount of water across the sides of the tube. And when we do that, it's through the process of osmosis. And I will stop here. I want you to review the isotonic, hypotonic, and hypertonic. This is where we'll start next time for the rest of this week. So this one chapter is for this week, and chapter 6. And if you can watch the video and do the worksheet, next week we'll move on to the next chapter 9 and get into that. We only have four weeks, I think, until we're done with things. So I figure this is a good balance, a good chance to boost your grade and learn some stuff. And if you have questions about anything, just send an email, or you can bring it up in a office time that I have with Zoom. So I hope this helps, and I can't see you, but it's nice being able to communicate with you. And again, I hope everyone is well and is doing what they need to do. So until the next video, be safe.